Okay, let's uh, let's get started. People continue to filter in, I expect. So it's you ready, John? It's a real pleasure to introduce Dr. Jonathan Loree today, who's going to be talking about um, the CCTG, or the Canadian Cancer Trials group, to let people know how one might um, collaborate or get involved with the CCTG. Um, I've known John since he first arrived here, I think, where we were meeting about a couple of projects. And before I met him, I was told that he was a superstar, and so I was a bit. It was with temerity that I attended the first meeting, and he is a superstar. He's done some incredible work. That just had a paper come out in Nature Medicine. John did his medical oncology here at BC Cancer, and then went to the not Memorial, so went to MD Anderson before coming back here. And his main research interest is in biomarker discovery using ctDNA in clinical trials in colorectal cancers and neuroendocrine tumors. He's the USNCI Colon Cancer Task Force co-chair, a member of the ASCO Evidence-Based Medicine Committee, and is the co-lead for the Correlative Sciences and Tumor Biology Committee at the CCTG. And he's a very pleasant person and great collaborator. So thank you, John, for doing this, and looking forward to hearing what you have to say.
All right. Well, thank you, Ali, for that introduction, and thanks everyone for joining today. So I'm a medical oncologist here in Vancouver, but I also have another hat at the CCTG, where I'm a senior investigator and lead the Correlative Sciences and Tumor Biology Committee. And I think, the CCTG is something that most of the trials that function across our Canadian sites run through, but a lot of people don't necessarily know kind of how the CCTG is set up or how to engage with. And I think when it comes to collaboration, one of the biggest things is knowing where the door is to knock on, and how to get engaged. And so I'd like to kind of give you a bit of background about the CCTG, talk about some areas that we think there's opportunities to work together. And I think the CCTG has really realized the importance of the pathology community in the success of research. You are essential to the success of clinical trials, both for the trial that's accruing today and for the correlatives and hopefully the ideas that get generated for future trials. And so we really want to engage, hear about areas that you're struggling with, and strains in the pathology departments, ways that we can streamline and work, be more efficient and make things better. But then also highlight some opportunities, within our trials, that there might be spaces for your research platforms and the things that you're interested in asking questions around. These are my disclosures, mostly related to development of clinical trials and the drugs that we test in those trials. I'd like to start with a land acknowledgement and acknowledge that I live and work on the traditional, ancestral, and unceded territories of the coastal Salish peoples. That's the Musqueam, Squamish, and Tsleil-Waututh, for where I live and work and play. And just acknowledge that there's people coming from around the province and different custodians of the land in those areas. So take a moment to think about that. As I mentioned, we're going to be looking at CCTG. I'll give you a bit of background about structure, how a trial gets operationalized, and then we'll review some ways of getting involved and different opportunities and areas that we'd like to hear from you as well. So the CCTG was just established in 1980. This was something Joe Pater started, and there's kind of some forefathers within CCTG who are still around. It's supported by the Canadian Cancer Society, so that's a big philanthropic group that gives a core grant every 5 years. It's renewed, and there's a big grant that goes in to set priorities and evaluate how things are going, and what might need to change, and where kind of to focus things. There's other supports coming from different philanthropic organizations, grants, as well as industry support, particularly if there's drug X or Y coming from company A, and that those funds help to support those trials. it is a bit unique in that it is kind of a full spectrum of resources. So it's, phase 1 through 3 trials, but then there's also committees that focus on quality of life, correlative sciences, there's patient representatives, there's kind of all these different pieces that come together to try and give a really wholesome assessment. And it's not just about assessing does a drug work, but trying to look at drugs and questions that we have that drive and improve patient care in the Canadian system. And we want not just drugs to show a 6-week benefit, but we want drugs that are, we can show the benefit, we can show the economics work. So there's a cost utility analysis for all the trials. It's kind of all these pieces to make sure that we don't just do research and not have it translate to the clinic, but that translation into the clinic is really important as well. So the mission of the group is to develop and conduct clinical trials aimed at improving treatment and prevention of cancer. Most of the trials are really in the treatment realm. There have been some collaborations across prevention, and I think that's an area that there's certainly some interest in trying to pursue more into. And most of the trials through the CCTG are adult focused, but there are also some collaborations with the pediatric groups. As I mentioned, there's phase 1 through 3 the whole way through, as well as all the research around that. It is a pretty extensive network, so that includes over 2,300 Canadian investigators, over 80 Canadian institutions, I believe it's 85. And then there's 500 international centers that have been spread across 40 countries participating on trials to date. And it's a bit unique in that it's the only cooperative group outside the US that's part of the NCTN. So this is something that's funded by the NIH, so groups like SWOG, NRG, ECOG Akron, and CCTG, they're kind of all part of this. And you can, there's a very structured mechanism of proposing a trial, getting it funded, and having it operationalized across all those sites. So CCTG has been lucky to be part of that currently, and there's some questions about the future of that, with the current situation, but it has really led to a lot of really good, strong collaborations there. There are over 170 trials running concurrently, so it's not a small group. It's a lot happening, over 111,000 patients enrolled. It's usually about 1,500 to 3,000 patients are enrolled onto trials per year, and that's led to a lot of production, so over 2,200 publications to date. And there's these active trials in the data repository, but there's also a biobank. It's called the TTDR, or Tumor Tissue Data Repository. And so from all these trials, there's samples that are banked. Some of those were pre-specified analyses. Part of that might be for regulatory filings, validating certain biomarkers associated, but there's also pieces that live on forever and can be accessed by researchers in the community to ask subsequent questions. And that can be blood, plasma, urine. There's sometimes, if there's a derivative like DNA is extracted from tissue, we'll get the derivative back. So that way that can be used in the future. And it's really meant to be kind of maximized so the most can be done with it. CCTG is kind of a virtual organization plus a physical structure. So there's the operations center in Kingston at Queen's University, and then sites spread across Canada. So it is really all across Canada. The Facility Management and Operations Center in Kingston, the structure there is you've got faculty and senior investigators. So these are individuals like myself, Crystal Callahan, Janet Dancy. These are people who kind of have an area of focus that they lead. So like Chris is the GI head senior investigator. Janet leads melanoma and is the director. Leslie Seymour leads the IND program. So that's like phase 1. And then there's senior biostatisticians that have different parts of the portfolio that they cover. There's trial management teams. That would be people like study coordinators who will actually do the day-to-day kind of operationalization. Patient's going to be randomized. The sample's not here. Let's get the sample, all those pieces. There's an entire finance team that does things from budgeting for clinical trials. So, what's it going to cost to put a patient on, all the different components of drug distribution, all those pieces. They also help with finance for correlative studies. They can help with grants. What's it going to cost to pull this many slides from the TTDR or those blood samples, those types of pieces. There's a huge compliance office. So a big thing when we're running phase 3 trials is we need to think about running them with high quality. So that way they meet regulatory bars across not just Canada, but the US, EMA, all the different regulatory compliance that's required for that. And in particular, because many of the trials actually run across all these different regions. So then we have to be able to incorporate the regulatory piece, differences in ethical consent, and what are the different rules in different regions. It's really important. This is all enabled by a strong information technology group that has a lot of online tools that help with all this. There's a lot of in-house developed databases and other pieces. And I mentioned the TTDR is that biobank that lives in Kingston. These are the member sites spread across the country. So you can see it's very diffuse. There's patients really spread everywhere, except for the territories. We don't have good representation. We haven't really opened trials up there. A lot of those patients get their cancer care at affiliated centers in the various provinces, and so they can go on study that way, but that's certainly an area that we haven't necessarily had an active site per se. The sites have a 5-year contract, which is really helpful from an efficiency perspective, because that contract is really essential to have the trial be able to operationalize and quickly come to place. And so a site can join any of the studies that are kind of listed within the stable. They basically, all the tools are online for them to join, and they kind of self-nominate that they want to join that study, and then they can be part of it. And this 5-year contract is really essential for kind of helping navigate both that trial piece, but also things like how's the tissue handled and what's kind of the custodial status of that. And that's all part of that contract. There's a director, so that's Janet Dancy. There's something called the Clinical Trials Committee. So this is what approves which trials move forward. Once they're approved and they're actually operationalized, the DSMC, there's kind of one DSMC that runs over everything, and that's looking for safety, any issues with the trials that are ongoing. And then there's specific scientific committees that have different priorities. These are the committees. So you can see it's kind of intuitive as you'd expect. You've got a breast committee, a GI committee, a GU committee. So these groups meet, they come up with ideas, they propose those, they have oversight over their own trials. And then there's some endpoint committees. These are important because they spread across all the different disease sites. So quality of life, economic analysis, and correlative biology. Those are endpoint committees that you're welcome to join. The correlative science in particular, would love to have more people joining that and interested and engaged. It's a really good opportunity to hear about what others are doing, but also to access resources for your own program. And we do operate on, again, it's through the Canadian Cancer Society. There's kind of a 5-year strategic plan that we follow. This is the current plan. You can see not really big surprises, understanding cancer biology, reducing burden, and demonstrating value. And I think that value piece is a bit unique within the CCTG compared to some of the other cooperative groups. So the value is a really big piece in making sure that we have all the data elements in there to support health technology assessments and all the pieces that are needed to get it into clinic. There are some platforms that are priority at present. So cell therapy, there's a national cell therapy program, data science programs that's looking at trying to, if we do correlatives on a study, making sure that the genomics are available to others afterwards to look at. And pre-op and perioperative trials is a current priority as well, which really is important to tie into the pathology community because some of the endpoints are going to be pathology endpoints, complete response rates, things like that. Many of the trials have been quite high impact. This is things like adjuvant therapy for lung cancer, erlotinib for lung cancer, cetuximab for colon cancer. And then there's some supportive kind of questions as well. Things like physical exercise reducing the risk of colon cancer recurrence. That was a New England article this spring that was led by the Canadian Cancer Trials Group. So these are things that certainly impact our patients. Studies can be conducted within Canada, but we do often work with other partners in Canada. There's kind of those set sites I mentioned, and then there can be initiatives that kind of span where the trial and maybe the correlatives are worked on kind of with different groups. Across North America, we tend to work with the NCTN, so the US cooperative groups. There's something called the Cancer Research Institute that can also fund research. And then depending on the history and the shared interests across a particular disease site, we might have certain partners that we work with. So for example, in GI, we tend to work quite closely with the French through Unicancer or Prodige, as well as the Australians through AGITG, and different groups might have kind of different key partners. A lot of these partnerships are driven by kind of shared healthcare systems, shared priorities, as well as relationships that develop. You have one successful trial and that leads to many others. A trial can be operationalized within Canada, or there's some different models. There's the intergroup models, that would be like CCTG working with AGITG, which is a group that works in Australia, and they kind of oversee their sites in Australia. We can also have sites come online where they're a single study CCTG site. So the trial can be open in Canada and then select sites in the US or England or wherever it may be. And those sites kind of become a CCTG site, and we do all the monitoring, all the aspects for that particular trial. So that's called the international model. We work closely with industry, obviously, because it's where drugs come from. That can be study by study, so there can be a contract for a particular study, and sometimes it can be a multi-study collaboration. For example, AstraZeneca had their Durva and Tremi kind of portfolio being developed about a decade ago, and there were a number of trials across all different indications that kind of rolled out all at the same time. We have worked with over 25 companies over a 40-year history. The reason for collaboration is really, it's access to CCTG investigators and that expertise, an ability to, potentially run a trial uh, in a unique indication or different questions. Sometimes a trial might be done in the US and then we don't have it approved, but it might actually lead to a real unique situation where we have an opportunity to generate some different data. Trials can either be in the IND portfolio. So the IND portfolio is kind of the phase 1, phase 2, earlier drugs. These can be approved on a monthly basis. They have a phone call basically once a month where they're going through different concepts. If it's agreed upon to do the study, a protocol to first patient in is expected in kind of 2 to 4 months, so pretty rapid. These are often kind of early drugs. We want to get a signal of safety and move on to something else. The phase 3 program is a little bit bigger because we're talking about regulatory compliance at a very different level if we're talking about a registrational study. Those studies move through something called the Clinical Trials Committee that meets 3 times a year to review proposals, and then those can move into operationalize from there. So how's a trial born before we get into kind of one of the pieces from a pathology perspective of how do you get involved and what are some opportunities there? So anyone can have an idea. If you have an idea, you can bring it forward and you can propose that concept. It usually goes to the disease site that it's specific to, or we can help find kind of a home, and they usually have monthly calls. It gets kind of workshopped there, and if on that monthly call people say, yeah, this is a good topic, then basically a senior investigator or maybe a statistician would work with you to develop that. And then it would be brought forward to either a spring meeting or potentially straight to a CTC if it's something that the timing is off as far as the spring meeting after it's been refined. And then if it's approved, then there's help basically for a couple of different things. If it's a study that you're coming with money already from a drug company, then they help with the contracting. If it doesn't yet have funding, then they would help with a CIHR grant submission or wherever the grants may need to go. That can be help with things like budgeting. Can be helping with understanding some of the logistics and making sure that there's not pieces missed as far as distribution or kind of hidden costs, things like that. And they really have a very strong grants team to help with supporting that. And I would say a high success rate in that place. So I kind of look at those as situations where you get a hunting license to go get the funds to do the study that you want to do, and they are a strategic enabler to help make that happen. Funding can happen in a number of ways. I mentioned the Canadian Cancer Society, the NCI. There's industry as well as these other granting agencies. And then once we've got the funds in place, there's kind of a specialized ops team that helps to make that happen. And so these individuals have lots of expertise in their kind of particular areas. So a senior investigator is kind of the medical and science lead. Got project managers that help with kind of getting it off the ground, people who help with the Health Canada submission or any paperwork in that sense. There's statisticians, programmers that help with the database, all of these aspects, they all come together to help make it happen. It all has to happen on a backbone of SOPs and well-documented training to make sure it's done at a high level of quality, but also so that way it meets those regulatory bars down the road. And so this trial essentially moves from protocol, contract being finalized, there's regulatory approval in each country, central activation, local documents, and then local activation at a site. While the trial is running, there's a monitoring and auditing program that CCTG does themselves on the sites. That's based on the risk of the study. It depends on if this is a novel drug with lots of potential side effects, and side effects are monitored because there's certain side effects that are going to be reportable. So SUSARs and other side effects that are serious and unexpected might have certain obligations for us to tell someone like Health Canada or the FDA that, hey, there's a problem that someone got really sick on this drug. And this is all enabled, again, as I mentioned, by that information technology group that's there that sets up all these databases and kind of all these online tools that are in-house built, which help to facilitate research translation. Now, what are ways to get involved? I think one of the really important pieces is CCTG has acknowledged that your community is essential to the success of clinical research and clinical trials within CCTG. We had a spring 2025 pathology workshop where we brought together pathologists from across the country, asked about what are the barriers to engaging in clinical trials, like what costs are not being borne, what are the human resource implications, what are ways that we could try and make things easier. And thinking about ways to work together. There's a white paper that's being developed that's going to be circulated afterwards. And it really came down to 5 key themes. So engagement of pathologists early in trial design, making sure that there's not a correlative manual that goes out that asks for something that's ridiculous and not possible to do. Having feedback metrics. So knowing kind of how many of the patients that are going on a study actually have the samples sent in that are supposed to be sent in, and thinking about ways that compensation can be a little bit more harmonious over the years instead of per patient funding. That's difficult because trials generally are per case funding, and that's the model that's very difficult to change, but thinking about ways that we could advocate for that together. We identified digital pathology as an enabler that we need to work on. Another piece was recognizing the contributions of the pathology community. So thinking about, when it comes to promotions or authorship, how are ways that we identify the pathologists that have been involved at each of these sites, and how do we get them credit for what they've done? And then also helping with knowledge translation around the actual ethics governance around stewardship of samples, how that differs in each region around the country, and how do we harmonize that. After the workshop, we do still have some pieces that we want to think about. We want to have kind of a resource that looks at what are the actual stewardship pieces, making sure that we're doing something that everyone is comfortable with. And if that is an issue, why there's not samples for certain trials, how do we deal with that? We're considering a pilot of decentralized banking of samples, because right now we ask for samples to all come in at the beginning of the study to make sure that they're there when we want to do the analysis later. But maybe that's something that we could save for only when the analysis is actually being done. That was feedback that we received from the pathology community. We're also going to capture on all those studies moving forward that site pathology involvement, and how we do that is a little bit something that we're working on developing. And being on the delegation log for a trial is a little bit different than just having the name associated with it for what training might be needed, and some of it might not be relevant to the pathologist. So we want to make sure we capture it, but do it in a way that doesn't just make extra work. And then we're going to plan for CCTG representation at Canadian pathology meetings and sessions such as this. Thank you for that invitation today. So getting involved, what are some different options? The first one is becoming a member, which is pretty easy. If you go to the CCTG website here on the right, on the far right, you can click here, become a CCTG investigator, and they'll take you to a website where they've got information. You can sign up. It's free. They don't send a lot of emails or anything like that, so it's pretty low burden. And that's kind of the first step. If you wanted to join a committee, when you're on that main website, in the top left, there's an About Us category, and then you can look at the different committees and see if there's something that appeals to you. And you can also find the committee leadership. So if there's someone you want to reach out to and say, hey, what's going on in GI portfolio? This is my interest. I'm interested in getting engaged. These are individuals who can kind of point you in the right direction. And you can also join the committees through this avenue by either emailing the SI associated with that group or the disease site chair. So for example, if you were interested in getting involved on the CSTB, the Correlative Sciences Tumor Biology Committee, you can just reach out to myself and I'd get you on the email list. Again, there's not a lot of emails. Most of the stuff happens around spring meeting. We are going to have some seminars coming up in the spring where we talk about just trying to raise awareness about what samples are actually in the TTDR, how you access them, and then what they can do in-house. And then hopefully we'll have a kind of a roadshow of the different core facilities around the country, so you know kind of who you can go to if you have a project that you're interested in. Once you've joined a committee, attending the spring meeting is probably the next best way of getting involved. I've got the dates there on the right. So on the main web page, you see meetings and education as a drop down. They've got the spring meeting of participants. There are funded slots for certain committees, as well as each site gets a certain number of funded slots. And then for those who are unfunded, there's kind of a special event rate. The attendance, I don't think the registration fee is as much. Maybe it's like $20 or something. It's not a huge amount, but there is still potentially travel to Toronto if it was an unfunded slot, so you're aware of it. But there's a multi-day, so it's basically a Friday and a Saturday, usually in April. There's a bunch of different sessions. You can pick and choose what you want to come to. It's a little bit more of a working meeting than a conference. So like in the GI session, there might be, new trials being proposed in colon cancer, and we workshop those and kind of vet whether they should move forward. In the CSTB session, our plan for this year is to have half of it be around ctDNA. Particularly, we're going to kind of pick a couple key topics, and whether that's in organ preservation for different cancers or other kind of key topics. We want to really focus it down. There's often invited international experts that are also there. And then in the second part, we're going to talk about ways to kind of future-proof correlative analyses, thinking about IP, how are you collecting samples, things like that. And then if you have an interest in a particular topic that you say, hey, CCTG should really do this, we do have these special workshops that we can set up at that spring meeting. There's funding to invite a bunch of people and bring up a topic. So last year, David Schaefer said, pathology, we need to think about ways to make it CCTG work better with the pathology community. And we had a pathology workshop meeting where the night before the before the actual annual spring meeting, we brought in a bunch of people from around the country and developed this white paper talking about different ways that we can try and make things better from both sides. Getting involved from a local perspective, we do have lots of clinical trials that are always moving through, and they need operational approval, and they need pathologists to review and think about what are the challenges with this for our site, and is this something that's going to happen? And so, being open to studies at your site is really helpful. There are some that require a little bit more testing on site, such as the EN11 trial. This is a gynae study that's looking at particular molecular subgroups that are defined, and that allocates patients to different therapies. So that can be really informative in a way for us to move things forward, both locally, hopefully when these things become standard of care, but also on the research front. And being part of these also gives you an opportunity to propose correlatives in the study once it's done, once it's fully recruited. And so we're going to be piloting a number of ways to recognize those types of contributions. As I mentioned, having a certificate you can use for promotions, trying to do a better job of capturing more authors, group authorship, other ways of getting more people on there, and then thinking about those future correlatives. Just to highlight this is the CCTG portfolio, the number of patients that are recruited by these disease sites. And this is a proportion of patients with no samples received, where it was in the protocol that a sample, like archival FFPE, should be banked. And this is the proportion of patients that had slide sense instead of FFPE blocks. And we're certainly seeing a transition in that. More and more sites are just sending the slides. We understand there's lots of different implications around that, but it does get a bit challenging when we're talking about long-term nucleic acid stability. And so that's part of this idea of decentralized banking. Maybe we can do something to make it so it works for sites as well as for future investigators thinking about their project. And then correlative proposals. So once those samples are in the bank, on the main page, so above Become a CCTG Investigator, you can click on Tissue Bank right there. And on the Tissue Bank website, there's actually an inventory that you can go through. And when you look at that inventory, you can see, this is an example of pulling up CO21, the study, and you can see there's over 1,000 patients that were accrued. And then down below, it would say the number of patients who have an FFPE block or slides or blood samples or what's available. And so this is being updated right now. We're going through renovation at the TTDR, and we're upgrading the computer system as well. So it'll be a little bit more real time, so you can see exactly how many mils of plasma is available at each time point. But these are things that you can propose studies to if you're a member of CCTG, and you can say, I want to look at protein X or Y or this mutation in this sample. And so it's a really good resource to try and ask your questions. It's really not for generating kind of your first pass of data. It's more, these are gold level clinical trial samples. Many of them have a comparator arm that's either placebo or best supportive care. So fantastic for looking for a predictive biomarker because you can do a test of interaction. But it's kind of once you've already got all your preliminary data, trying to validate it, then here. The CCTG biostats team helps with the analysis as well for this. If you say, I've got this cut point and this is what I want to use, and here's all the preliminary data, then they can run a lot of that analysis for you. And the proposals are pretty straightforward. This is what the proposal looks like. It's, kind of contact information, how much you need of things. And then it's a 3 to 4 page document, which is kind of like the protocol you'd have at your REB anyways. Brief intro, hypothesis, objectives, background, preliminary data, methods, stats design, significance, and why you need clinical trial material. And these are reviewed on an ongoing basis. Usually what happens is they get the trial team that led the initial trial to review and say, hey, actually somebody's already approached for that, or we did that actually 10 years ago, because many of these trials can be quite old. And then there's often an expert kind of in the field, whatever that particular field may be for the correlative, that provides some feedback. So lots of ways to get involved. I think the first one is just becoming a member. And if you're interested in joining a committee, I think the CSTB is pretty well suited. If you're open to coming to Toronto for the spring meeting, I think it's a great way to network. There's tons of investigators from across the country there. And then proposing special workshops if you have an interest there. Certainly happy to help navigate that. And then if you get involved in trials, that's both kind of thinking about contributing to the protocol and subsequently the correlative analysis. It's a great way of getting access to kind of these gold cohorts. We can do a lot with a TMA and local questions, but then once you get to a point where you really want to validate something and take it to the next level, these are great opportunities. And there's a lot of tissue that's available there. And we can also access the tumor bank at the NCTN. And so that's from all those NIH clinical trials that have been run in the past as well. So hopefully that gives you a bit of a sense of CCTG, the structure and oversight, just so you know who and how to engage with, and then thinking about some different ways to engage. And I've left lots of time in this, so that way we can talk either about past experiences that you might have had, or about either suggestions you have for CCTG for how to improve, or if you've got questions about different ways that you might want to engage with the group. So thank you.
Thanks, John. That was fantastic. Nice to see the veil being opened of how CCTG operates. That's fantastic. So, do we have anybody have questions? David?
Yeah, John, you alluded to the the political situation in the States, and we know like funding priorities have changed there. How has that impacted the CCTG, or perhaps even created new opportunities? Because usually when something bad happens, there's an opportunity to.
Yeah, great question. I think the biggest area of uncertainty is that we currently receive some of our funding from the NIH through a grant, and that's kind of under review at present. And we don't really know the outcome of that and our future really kind of relationship with the NIH. I think regardless of whether that gets approved, there's still lots of ongoing relationships with those US cooperative groups that will be maintained, but it might be a little bit more difficult to work together in those. I think it has caused us to reach out even more to some of our international partners and strengthen some of those relationships. So for example, we're in colon cancer, we've got a big phase 3 that's going to be Canada, Australia, New Zealand, and France. And that's something that, before there might have been more discussion around working with one of the other cooperative groups in the US. And I think it's kind of caused us to be a bit more diverse. I think, at ESMO this year, which is our, we kind of ASCO in Chicago and ESMO in Europe as our big medical oncology conferences. You could see there were a lot more people, a lot more of the big studies were presented there. And I think there was a lot more folks from ex-US countries kind of looking to collaborate and work together. So I think you're right. There are some opportunities there for us to look out and find new partners.
Thanks.
Anyone else?
Yeah, Cedric here. The tissue bank obviously has a limitation on the amount of sample. If people apply to you, do you have an adjudication committee on how you can allocate these samples?
Yeah, it's basically a mix of the trial management committee for that initial trial, as well as some members of the CSTB that would review. And it's based on kind of, what else is planned with that trial, how much, where it's at in its lifespan, and then kind of the preliminary data for whatever you might be proposing and kind of what you're bringing forward and the feasibility of it. If they're very good about saying, well, actually there's not much FFPE left for that, or we had issues with tumor content before and on this set or what have you.
Thank you.
Dr. Gao.
This is Gao here. Well, a very nice presentation. Thank you. It certainly opened my eyes for more opportunities. Just a very quick question for those tumors that have been utilized in the trial in the bank. Do you have like scanned slides for all those in the database?
Yeah, great question. So it's becoming our standard kind of moving forward that we scan every case that comes in. So that way we just, as a matter of practice. It wasn't previously the method or the kind of practice. And I would say that we do have some initiatives and collaborations that are facilitating us going back to some of the older studies and scanning them in and kind of building up those data sets. So obviously machine learning AI projects, there's a lot of interest in that. And it's nice to have a resource that's not depleted for a proposal. So.
Thank you.
Would you, John, would you clarify, well, I don't know about clarify, but just maybe flesh out a little bit more about samples that you need. You talked a little bit about the issues, but sort of what's, besides what you presented, what do you think the way forward is here?
Yeah, I mean, I think it's hearing from sites of kind of what is possible and what they're open to. We're hearing more and more that sites are just not comfortable sending blocks. And I think that really depends on the case, whether I think that's something that makes a lot of sense. You know, if it's a resected colorectal cancer that has upfront a lot of diagnostic blocks, that's very different than, you know, our metastatic pancreas, where there's a very small amount. And so I think trying to work to engage pathology early about what's pragmatic for those trials. And then one of the big pieces is making sure that it's the pathologist that says that's our policy, and not just kind of someone who's in an operations role saying, well, no, we can't send that. I think that's kind of an important piece too. And that's to make sure that the resource is something that lives on for the investigators on the study, but also for you as the pathology community. Like, you don't want a bunch of slides that were cut 10 years ago if we could have a block that's sitting there waiting for you. I think that we're going to try this decentralized banking, so that way hopefully that's not an issue. But I'm worried that we're not going to get the blocks down the road, because obviously everybody's strapped from a human resource perspective. And if it comes to doing a correlative analysis from a trial that you have to pull from the archive as opposed to some clinical work, clinical works can get prioritized. And I think those projects are going to have more feasibility challenges, but we'll see. And I'd love to hear if there's any suggestions from you guys about ways to make it so we can make it pragmatic, but we get wholesome samples that are going to be useful in the future.
Yeah, when you say decentralized banking, are you suggesting that the blocks from the trials be held in a separate?
And the idea is you would keep them somewhere special on site where you could pull them. You would not send them out to Iron Mountain, but you would keep them on site, so you'd still have custodianship. And so we said specifically, yeah, now we're going to extract and run the RNA-seq on it.
Sure. And there's funding that would be that would come with that?
Well, that's a great question, because right now we manage that all in the core. We hold on to them for that length of time.
Yeah.
What do you think that would take cost-wise?
You know, I think it's going to be institution dependent.
Yeah.
Yeah. And maybe even, yeah. I'll leave it at that.
Yeah.
Yeah, I mean, an advantage of the model you describe is it would be akin to federated learning systems, where people host their own data as well. So it's an idea people are becoming a little more comfortable with.
Yeah. I think I worry that the samples aren't going to be there in 10 years when someone says, oh, I've got this great idea. And so there's kind of...
Or technologies appear that you can actually ask the question that you couldn't ask before.
Yeah, that's true.
Like ctDNA, right? Nobody
Yeah.
saved plasmas back then. So, and then what about, speaking from personal interest, what about hematologic specimens, liquid tumors?
Yeah, I mean, they're in there. I don't know for each trial what they've got, but for any of the hematologic samples or trials, they would have also been banking, but kind of depends on what was left over. Obviously, it's a little bit harder to get a bone marrow aspirate than a big juicy resection from something else.
Yeah, well, we've received some of those samples, but I guess I'm just thinking about, you know, a federated or decentralized model. How would you, because that's going to cost, you would need liquid nitrogen storage to store cells viably.
Oh yeah, for the storage. Yeah, I think there's that. And then there's also the making sure that after someone's run an assay that we kind of get it back and we have it, you know, it visible. So that way people know they don't propose something that's already been done, but also so that way, you know, if you go and do an exome, and Bob wants to look at something very similar or different topic, then he can use your exome. And I think that should be part of it, part of the kind of commitment to, if you're going to use those samples, that kind of once you're done with your question, you give the data back in a way that others in the community can use.
Yeah, what identifier are there identifiers you can use that would be acceptable to put into public databases? Is a question we've been talking about internally, because we upload all our data.
Yeah.
The question is, how do you, I mean, they're de-identified, but then how do you link back to the clinical, maybe more extensive clinical data that's on site?
Yeah, so like CCTG doesn't have personal identifying information in a way that you'd be able to link to a patient in BC Cancer. So the code, like if a patient's enrolled in Vancouver, they get CAVA, C-A-V-A 001. And we get some information like month and year of birth, those types of things. And then there's specific dates once they're enrolled on the trial, but like you wouldn't have the patient's PHN at CCTG, like they wouldn't be able to link back. So obviously you could do a little bit of digging and maybe get kind of close, but it'd be pretty hard to link it once it's in the CCTG realm.
Right, but you could use that identifier so that CCTG would not have to host all that data, but you could go back to other repositories like EGA or NCBI and
Well, I think if CCTG were to upload it, then we would link it to like CAVA 001, and that's considered de-identified. The biggest thing there would be if the consent in the first place allowed whatever the type of data sharing agreement. I think we've learned, because there was an interest in having more kind of Marathon of Hope type cohorts within the clinical trials. So now the consents are a little bit broader for that kind of data sharing initiative. But there have been some historically that maybe have a little bit more restriction on the data sharing piece.
Yeah. Thanks, John. Are there any more questions? All right, great.
Thanks.
All right, thanks
very much, John. Great talk. And you already invited people to reach out if they want, so I'm just reminding people of that. So thanks a lot again. Take care.
Thanks for having me. Happy Halloween.
And to you. Oh.
Thanks, John.